Good morning, our student. How do you do? Precisely, we have studied both types of nematodal parasite that dwell liver and lung in the preceded lectures. Today we will talk about other different species of nematodal parasite that can infect our GI, gastrointestinal tract. Today we have two species. Now, about the intestinal flak. The first species here is Pathogenicity yakagawa, as a scientific name, Pathogenicity yakagawa. Its common name, heterophyte flux, and can cause metagonomiosis. Now, about its morphological feature of the adult worm. This parasite is a smallest flak. Here, a smallest flux is Pathogenicity yakagawa. To affect human and other mammals such as cat, dog, and pig. The worm is piriform in shape, with tapering occurring at the anterior end and surrounding at the posterior end. Okay, a thin layer of scaly spines cover the organism, which is heavily distributed over the anterior end. This parasite can live in the small intestine, as we mentioned before, of the definitive host, we mean human and other mammals, can be found within its GI, at the villi. Now the ventral suckers is characteristically deflected to the right, with its long axis in the diagonal plane. Now here also this slide can show you the morphological form of stained slides of Pathogenicity yakagawa, the smallest nematodal flak. On the left, stained slide and of the adult worm stained with the carmine stain, whereas at the right side of the slide we can see the adult stained, and also can see the egg, two stages, up the adult worm and beneath it the egg stage. Other slide. This slide deals with the egg. This egg is consisting with a developing parasitum. So the egg, when be hatched, it having developed, embryonated, we mean it's embryonated, not unembryonated like that studied preceded in preceded lectures. So it consists of developing parasitum and except an operculum. Yeah, it is operculated. Now it is resemble that of Clinorchus sinensis and Paraganimous ceremony, but can be confused and indistinguishable. There are only two somewhat discrete differences among the egg of Heterophyte and Pathogenicity yakagawa versus or opposite those of Clinorchus sinensis. Here is very important criteria. Two differences that can resemble the egg of Heterophyte and Pathogenicity yakagawa, which is different from that found in the Clinorchus sinensis. The first is the Heterophyte and Pathogenicity eggs have shoulders. Okay, it's half shoulder like that. But they are with less distinct than those of Clinorchus. The first difference. Second, the eggs of Heterophyte and Pathogenicity may lack the small terminal lobe found in those of Clinorchus. Sometime or may lack not prominent, may lack, cannot be void. Okay. This slide can show you the life cycle of the Meteor Sakagawa. Now, a human infection as a definitive host and other mammals can be infected. How the infection can occur of both Trophes and Meteor Sakagawa can occur after ingestion of metacercaria present in raw or contaminated undercooked fish. The second intermediate host that harboring or affected by this parasitic pathogen after ingestion the infection can occur. The adult worms of both species reside in the small intestine, in our GI, the general region, especially. The mollusks and fish are the first and second intermediate hosts, respectively. Now the cercaria has a lateral floating tail. Okay. Other slide. Now here we can see the life cycle presented clearly here. Now in this shape we can see a whole life cycle which is similar of both Meteor Sakagawa and Trophes. Both species can dwell and effect present the adult worm can present in our GI, and the life cycle is similar, so similar, resembled. Now, on the right side of this slide, we can see the human how can be infected. Now, the human, we mean host, human or mammals, become infected by ingesting undercooked fish containing metacercaria. Now, the metacercaria exist in the small intestine, and then the adult will be formed in our small intestine, the adult worm. The adult worm can give what? Can give embryonated egg, not an embryonated, but embryonated egg, with its fully developed miracidium are passed in feces from human and from mammals, fish-eating mammals and bears. Now the embryonated egg can have two miracidium. The miracidium can enter the first intermediate host, which is snail. So the snail host ingest egg. Miracidium emerge from the eggs and penetrate the snail intestine. Now undergo several developmental stages. Now about its pathogenicity. The egg of this parasite is very important criteria. The egg of this parasite have the ability to escape in the lymphatic and venous via intestinal wall filtration and to migrate to other area of the body such as heart and brain. Now granulomas in this area often result. Granulomas or calcification can occur in this ectopic sites. Well, about its clinical symptoms, two phases, asymptomatic and symptomatic. The asymptomatic, we can say light infection, typically remain asymptomatic. The patient, the affected individual cannot be symptomized, not suffering from symptoms, not appear to be symptomized. Whereas in symptomatic or metagonomiasis as a disease, heavy infection, in heavy infection of Meteor Sakagawa produce abdominal pain and discomfort. Patients often experience a chronic mucus diarrhea and eosinophilia. It causes a catarrhal condition of intestinal and diarrhea. Well, another slide here can show you how can we diagnose it in laboratory and how can we treat it eventually. Now, laboratory diagnosis is, or it can be made by recovering the egg in stool samples. This is difficult. Very important criteria. This is difficult to achieve. Why? Maybe you ask yourself why it is difficult. Because the reason is because the egg of both heterophis and Meteor and clonocal sciences are so similar to each other. Okay. Now, about its treatment. Now, the treatment of choice for metagonimous infection is the praziquantel drug. Well, we will change or deal with other species in the lecture of today, which is heterophis. Now, our student here, the second species that will our GI by intestinal flux, other species of intestinal flux, which is heterophis, which can cause trophiasis. Now, about its criteria. The adult form can appear pear-shaped, very small size, 2 millimeter. Now, the egg is small and operculated, having an operculum colored with yellowish to brown with thin shell. Okay. Now about its suckers. There is 3 suckers, not 2 suckers, 3 suckers, which are oral sucker, ventral sucker, and genital suckers. Okay. Now there is vitelline gland, be void clearly here, and 2 testes and 1 ovary with simple intestinal ceca. You know, these pathogenic parasite or trematodal class of trematodal parasite be hermaphroditic. Both reproductive organs can be found with the same parasite, so there is no male and females not be separated. Well, now about the location of the adult womb, where is in a small intestine. The worm can be present in intestinal villi. About its intermediate host, there's 2 intermediate hosts, which are first intermediate host is the snail, and then the second intermediate host, which is fish. Now, the infective stage is what? Is encysted metacercaria that found in the vagina of second intermediate host, fishes. Now, how can it be modi to its transmission routes? How can be transmitted to cause disease? The mode of transmission is occurred after ingestion of raw or undercooked fish containing encysted metacercaria. Now you know the disease is called the disease that caused or mediated by such heterophis flux parasite is heterophiasis. Now we can diagnose it by examination of fecal specimens. Here other criteria in this table can show you the shape, the color, stage of development when passed, specific feature and variation. Now the heterophis parasite shape, we can say it is the smallest and small, elongated or slightly ovoidal. Now operculum egg. The slight knob at posterior end, which is sometimes be missed or not be detected, so it is difficult from clinical sciences. This is the second difference. Now about its color, the color, this worm can be appeared yellow to brown, and in this table must the criteria here consent to egg stage. Okay, now stage of development when passed from the definitive host, human being, it is embryonated. Eggs contain mature miracidium, and there is a specific feature and variation related to this parasite. It is a simple Clonorchis eggs, but with less distinct shoulders, upper canum. Now, is broader than any Clonorchis sinensis. Okay, these are criteria related to the Trophocytous parasite, especially the egg stage, and the difference, or which differ from that found in Clonorchis sinensis, that it was the liver. Now here the morphological feature of the adult worm, and the point here can show you the outer spines. The tegument of this worm can be equipped with the spines, cover spines. It's very clear and void. Now the oral sucker and ceca, it's very clear here, and there's the ventral sucker and genital sucker armed with spines. Now uterus, collet. Now the vitellaria and posterior third position, vitelline gland, and testes be rounded, 2 testes, and 1 ovary. Other slide. Also here we can see the stained slide of this pathogenic parasite, both in left and right. Other slide. Now about the morphology and life cycle and pathology and clinical feature and its distribution with laboratory diagnosis. All of these criteria or formation related to Heterotrophus parasite can be seen here, and other stages like Cercaria and ovum can be presented here. You know, our student, the adult worm shape here presented with its points and equipment, internal structures and surface structures. Now the life cycle can start it when the human discharge over to environment, and then over be embryonated, and the ovum can penetrate the face intermediate host as miracidium when hatch from it to enter face intermediate host snail organisms and to undergo sporosis, 3D Cercaria. The Cercaria leave the first intermediate host and enter the second intermediate host, which is fishes. Now after fish eating or ingesting such non-appropriated correctly second intermediate host for nutrition or fooding purposes, the human can get the infection, to be infected. Now the raw or inadequately cooked fish ingested by the final or definitive host, I mean human. Okay, now about its pathology and the clinical feature. You know, according to its site, be developed or infected, there is a mild inflammatory reaction Now the infected individuals can suffer or may be asymptomatic or have abdominal pain when be symptomatic, especially in the heavy infection, and there's diarrhea, can be anorexia and nausea. Now ectopic ova have been found in the heart and the brain. Okay, now it is found in the Nile Delta and Far East. It is with cosmopolitan distribution globally. Now the ova can be detected in the feces by examination for fecal specimens to be diagnosed in laboratory.